आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज, की आवाज।
Vigyan Ki Awaaz podcast series aims to popularize new ideas of science and technology, which highlight STEM, science, technology, engineering, mathematics, and medicine. This brings to the surface the efforts and researches of Indian scientists, which is desired to bring a change in community learning and teaching, access to knowledge of resources, and social paradigms. Our goal is to spread the name of indigenous Indian research stories and their masters among the masses of society in a very easy manner. Bose Institute was set up in 1917 by Sir Jagadish Chandra Bose, the founder of modern science in the Indian subcontinent. It is Asia's first modern research center devoted to interdisciplinary research and bears a century-old tradition. The institute was founded as not merely a laboratory but a temple, with its principal aims being the advancement of science and diffusion of knowledge. Swami Vivekananda, Sister Nivedita, whose influence is reflected in the institute's emblem of Vajra, was significant in the setting up of the institute, and Rabindranath Tagore were among those who had consistently inspired and supported Jagadish Chandra Bose in his endeavors. Ever since the establishment of the Department of Science and Technology, Government of India in May 1971, Bose Institute has been an autonomous grant-in-aid institution of DST. Over the last century, the institute has endlessly strived to preserve the legacy of its illustrious founder. In this podcast, we are happy to present Dr. Nirmalya, Assistant Professor in Bose Institute. Dr. Shen is working on cancer biology and found out a lot in curing cancer. But before going into more details of his study, let us listen from his own voice about his academic journey.
So, hello, I'm Dr. Nirmalya Sen, and I'm Assistant Professor at the Department of Biological Science, Bose Institute. And here I work on cancers. So, my lab is working on research with cancer biology. So, I'll tell a brief about my education. So, I did my MScs in Department of Biochemistry from Banaras Hindu University. And then from then, I went to do a PhD in National Institute of Immunology, New Delhi, and where I worked on tumor suppressor gene P53, which is a well-known gene to stop many cancers. So after doing my PhD, I moved to US and I did 2 postdocs, one in George Washington University in DC, and then subsequent postdoc at NCI, National Cancer Institute, NIH, USA. So during my PhDs, my work, I worked on coactivator of PGC1. So that's a well-known coactivator again, which is usually found during thermogenesis. That is, when you feel cold, this coactivator gets activated and it starts burning fats in our body. So how this coactivator was related to cancer, that was one of my work. So when a tumor grows inside the human body, so the core of the tumor starts lacking all those oxygen as well as nutrient molecules. Because if you see a tumor, it is growing like a sphere, like a round ball shape. So when the tumor is developing, it will need a lot of energy and a lot of food. So it will take it from the human. Now, as the tumor grows big, it develops a oxygen deficient environment inside. And due to that, it cannot even take good quality food. So at that point of time, these kind of coactivators help the tumor cell to progress. So in this case, P53 is a molecule, is a transcription factor. So transcription factors are basically DNA binding proteins that can bind to DNA and activate genes that may either help in certain functions. So in case of P53, it's also a master regulator because it is also known as the gatekeeper of cell fate. That is, when you have a cell, whether it is going to divide or it is undergoing arrest or it is undergoing death. So that all things P53 decides. So this coactivator PGC1, which was known in severing thermogenesis, that makes us feel warm during cold. So that thing can actually bind to P53, and from there it can regulate how P53 will cause death or life. So this was the work that got published into big international journals at that point, and from then I moved to US. So this was a work that was done in liver cancer at that point. So I moved to US and I found out that these transcription factors that I was working on, they can also join with other genes. So this is again called fusion genes. So this was a very new thing for me. So I thought, like, when you go to a postdoctoral study, then you should increase your experience. So I used my past experience of PhD and then I went forward to understand how these genes fuse. So at that point I was working in a childhood sarcoma, cancer of the muscle. So this is called Ewing sarcoma. So Sir Ewing found out the cancer in 1944, and now it is affecting mostly European kids, so like white background kids. So like 200 to 250 cases comes each year in US. So typically this affects male kids, not female. So we were working on one of those fusion genes called EWS-FLY1. So from there, I also collaborated with another group who were working with another child sarcoma called rhabdomyosarcoma. So this is again a muscle sarcoma which affects a lot of kids in the US. So we got back to have several publications out of it. We also found out what is the molecular mechanism that is undergoing and how to find out a cure. So we found out several drug targets there. So working with all those things, so some of the genes are common in various cancers. So today I'll be talking about the current work. So once I came back from US, I had a lot of experience there working with different transcription factors. So due to that, I started writing a project for the Indian government. So I got a fellowship called the Ramalingasamy Fellowship from Department of Biotechnology. It's a very prestigious fellowship. So I got that fellowship from the Indian government and I moved back to India. So these fellowships are given by government to restrict brain drain. So I came back to India and joined RJCB Kerala. So this is Rajiv Gandhi Center for Biotechnology. It's a very good institute in Kerala, again run by DBT. So I started a program there and I worked there for 1.5 years. At that time, I was working with a collaborative group from IIT Allahabad. So now it's called Prayagraj. So there we worked on pancreatic cancer. So they were basically a genomics group who doesn't do much of wet lab research. So there is a division. So they asked my help. So I collaborated. So it was a very good opportunity to increase my ideas about genomics and all. So later on, I started collaborating on a long term with that scientist. So we got a couple of publications from there. So after that, I shifted my job to University of Kolkata. So I wanted to get a flavor of state university.
Along with other scholarships, Dr. Nirmalya Sen has received Early Career Research Grant from DST, which is given to the young scientist. Dr. Sen is working on triple negative breast cancer. He has observed that one medicine is working on selective cancer patients. Let us know in details.
I started building up a lab. It was a bit difficult because state universities do not have much funding. So I did have my initial fund, and apart from that, I also got another fund as a ECR career grant. So it's called Early Career Research Grant from DST. So this is given to young scientists who have just started their research career. So that also I got, and this time I started working triple negative breast cancer. So the previous project was in prostate cancer and pancreatic cancer. So this time I shifted base to triple negative. Now, why did I choose these two cancers, prostate cancer, triple negative breast cancer? So nowadays, if you see, we have a lot of medicines. In clinics, if you go, a normal man knows like what is metastasis or what is a biopsy or what is a PET CT scan. So they know a lot of terminologies about cancer. Still, when you treat two patients with the same type of treatment, same type of cancer, one patient survives for 20 years, the other patient dies within say 1 year. Why this health disparity is there? So what I see is that one thing, the drugs that we are giving, some patient responds very well to them. So there is no relapse of the disease. They are cured. The other one is that the drug is not affecting properly. So then I went inside and I started reading what are the cases, and we found out that most of them are chemo resistant, that is chemotherapy resistance or hormone therapy resistant.
Dr. Nirmalya Sen explains how this happens, and mostly Indian women are affected by this disease and death rate is really high. Let us hear from Dr. Sen.
So as I have told you, like my whole life I have worked with various master regulators like transcription factor. You just tune them and the cancer stops. These regulators as a basic thing, these are also found in normal cells. So they do good function in normal cells. The only thing is when they get mutated or when they get damaged, at that point they start acting the devil's advocate, that they play the bad role at that. So in my case, I have seen that ETS transcription factor, a family of transcription factor, when I came back, I started working on that. These family of transcription factors started, playing this oncogenic role. So oncogene are genes that cause cancer. So they become oncogenic. So in case of triple negative breast cancer, we started collaborating with AIIMS, the newly formed AIIMS in Kalyani, West Bengal. So when we started, we saw that number of triple negative cases in West Bengal is too high. And we started going about India. We saw that compared to the Western population, that is the European and American women, Indian women are more affected by this cancer. The idea is to see how much our population is affected. We estimated it will be 25%, but now the current data from our lab shows it is around 40%. So out of 100 women, 100 breast cancer patient, 40% breast cancer patient will be triple negative breast. So again, coming to the divide of breast cancer. So there are various divisions of breast cancer, like there are various types of it. So triple negative is the most bad, like poor prognosis breast cancer, because they don't have the hormones called estrogen, progesterone and Herceptin. So the thing is, normally the breast cancer is treated in clinic using an anti-hormone therapy. So if you don't have the receptor, how will you give anti-hormones? So the only thing that clinicians or doctors are left with is chemotherapy. These drugs have various side effects, as well as these cells are getting chemotherapy resistance once you start treating. So women die very young, 3 to 5 years, and they are dead. So when we saw ETS1, one of the transcription factors we are seeing, we saw that this transcription factor is somehow modified once you give the drug. So it was evolving. The cancer cells was evolving into a resistant cells. So we thought like why it is evolving. We found out that one of those master regulators has changed. And now we also saw it in a clinical perspective. We collected patient samples. We saw that this protein has changed in the patient. So now we have a predictive biomarker.
Dr. Sen speaks more on the details about his findings and predictions.
So what we found out that this protein is phosphorylated, and when you are taking biopsy, if you use the antibody against this protein, we can actually detect this protein, and we can predict that this patient number 2 is undergoing chemotherapy, will develop a resistance. So we should carefully monitor the patient and change the course of drug. So this is how you can save a lot of life. So second objective that I found out is like we don't have non-invasive biomarkers. So if I ask a common question to a person like you have a cancer, how do you detect it? Normally they will say you have to put a hole inside a patient and take out some tissue, take out some blood, and then you do a test. It's a very painful procedure, FNAC biopsies, and normally in Indian context it is also a bit costly. So economically deprived patients, they don't go to clinics when they have cancer, and also they are afraid. Like if you put a FNAC biopsy, it will spread around the body and the cancer will be fast and I will die. So due to that, dilemma, they don't come to the clinics, they don't have an awareness. So I thought of developing a non-invasive biomarker. So you just take a blood sample, normally you draw it with a syringe that is not even painful, and then you analyze it using a paper strip kit. So for doing that, what I started doing is I collaborated with regional hospitals. I started collecting samples, triple negative breast cancer samples, and I started doing RNA sequencing. So I want to see what kind of RNAs is going up and down in those cancer compared to controls, like normal subjects. Doing that, we are trying to find out what are the secretive genes, like which comes out in blood or plasma or urine. So if I can get some of it in a kit, then I can develop a kit where I can just check in a cancer patient whether this patient is going to have a cancer in 5 months or 6 months without doing a biopsy. So this is where my future perspective lies, to modify different molecules that are being clinically used as drugs now to modulate these transcription factors as master regulators. If you can tinker them, we can cure cancer. And the second thing is to develop biomarker based kits. So that way we will have a detection technology. So these are the two things.
Dr. Nirmalya has given focus on the way forward.
One thing what I think nowadays is like in India, like developing hospitals like AIIMS, doctors are very much, coming forward, like we want to do something for cancer. So we need to, have a very formal collaboration with, clinicians and academicians and researchers. So if we don't collaborate, some of the skill sets I have, some of the skill set the doctor will have. So one thing is we should have good collaborations, and we should have the perspective to think good science. For that, government funding is there. So we are getting a lot of funds also for doing it. Another thing is awareness in people that cancer is curable. You should not be afraid. You should come forward. So we have cases now in India where people are approaching scientists and they are funding scientists to cure their, daughters or near ones or dear ones. I have heard a couple of cases myself, but I haven't personally solved those kind of things. But in near future, yes, this is one thing we are looking forward.
Dr. Sen also expresses his views on how Indian pharmaceutical industries will be benefited out of his area of research.
Indian pharma industries are doing both ways, like diagnostic is one thing. After COVID, you have seen a lot of companies are coming up with diagnostic kits for different diseases. So cancer will be one of the priority sectors for them. So definitely the identification of those kind of, biomarkers will help our companies. So patent them with the companies, one thing. And another thing is like some of the drugs are already there. So if you can detect early, then we can actually treat early those patients. So that way a lot of lives will be saved. So that is one thing I think will help us.
We are really thankful to Dr. Nirmalya for providing us with his valuable time and sharing the knowledge he has acquired through his area of research.